Hi, and thank you for that introduction. I have been with the agency for 15 years now, and I have had the privilege of working with a wide variety of companies and stakeholders on companion diagnostics. It is my hope that I will share with you today what I have learned along the way to support the success of your particular projects. Very briefly, my talk will provide an overview of the points to consider when planning the use of an investigational test in a trial. I will discuss common pitfalls companies frequently encounter and strategies to avoid them. And finally, I will discuss the regulatory processes for companion diagnostics. So perhaps the most frequent question we encounter from small companies and academic institutions alike is, why do I need a companion diagnostic? Isn't diagnostic testing part of the practice of medicine? The answer to that is yes, it is. But as you are aware, in vitro diagnostics are regulated by the FDA based on the specific use of the test, and more specifically, based on the risks to the patient if the results are wrong. This is an important point because CLIA oversees laboratory testing, and that oversight is focused on analytical validation, whereas FDA will also request clinical validation in support of the use of the test in a specific clinical setting. For example, there may be multiple reasons to measure alpha-fetoprotein, and the lab will confirm the ability to reliably detect and measure alpha-fetoprotein. But FDA will review the analytical performance and clinical validation for detecting AFP, depending on whether it is used to monitor testicular cancer, predict risk of hepatocellular carcinoma, or prenatally to check for spina bifida. And based on the risks of wrong results in each of those settings, FDA will determine the submission process. So companion diagnostics are regulated IVDs that provide information that is essential for the safety and efficacy of the therapeutic. The risks of this indication is that a wrong result may lead to inappropriate treatment decisions. The pharmaceutical sponsor is responsible for defining the trial population and for ensuring that the same population for which the safety and efficacy was demonstrated can be identified after drug approval. Both laboratory tests and commercialized kits are expected to comply with regulations when used to support drug approval because the performance of the test is critical and because the test information is going into the drug label. The drug will refer to an approved FDA test, and the diagnostic label will refer to the drug. So let's do a quick test to get us off on the right foot as we move forward here. Assess your feelings about all of this. Which of the following is true? Companion diagnostics have the potential to improve therapeutic efficacy. I think we all agree that targeted drugs increase drug effect by narrowing the population to individuals for which the drug is designed. Decrease adverse events. Similarly, by understanding the limited benefit or safety profile of a drug limits exposure to adverse events or lack of benefit. Support better quality of care and help reduce healthcare costs. Certainly occurs when you remove or limit the trial and error of exploring treatments. The savings, both financial and personal, with the treatment are of value to both physicians and payers alike. And finally, creates hurdles that cost time, resources, and money. So I think that this is frequently a concern that a lot of startups have, and yes, it can be true, but not if there is proper planning. It's important to note that FDA has created many initiatives to prioritize fast reviews of targeted therapies, and many large oncology drug companies have groups devoted to companion diagnostics because the clinical community and public recognize the benefit of these drugs. So market adoption is pretty strong. So if I'm able to convince you of one action to take with this talk, it is this. Action with this talk, it is this. Plan early. The drug and diagnostic submissions are intrinsically related. I am sure all of you have seen some version of this slide. It represents the ideal scenario for co-development of a companion diagnostic with a drug, where a single analytically validated test is used and perfected until the registrational trial. A final lockdown version of the test is used to select patients, and the market-ready IVD application is brought in at the same time as the NDA or BLA. However, the more common scenario is that companies are playing catch-up, and this is generally due to poor planning. Some of these scenarios, for example, include using a variety of local laboratory tests to enroll patients, and for this reason, there are no test negatives available to validate the companion diagnostic. It's also not uncommon to see early phase data used to support the drug registration, and now there's a scramble to get specimens and maybe even a diagnostic partner. The diagnostic partner or the pharma company decides to make changes to the test or changes to the cutoff during the trial or late in the trial, causing complications with the clinical validation of the companion diagnostic. Clinical decision points or cutoffs weren't nailed down during the trial, and so there's this ongoing investigation of optimal thresholds, leading to a need to retest the specimens. In other cases, safety concerns can point to a need for a companion diagnostic when none was planned. This occurred, for example, with several PD-L1 inhibitors. Patients were selected for a specific variant, or the intention was to select for a specific variant, but at the conclusion of the study, it was discovered that many more patients were enrolled with other variants in the gene due to the use of multiple tests. But the diagnostic test that's planned to come in as the companion is not designed to detect any of these variants. And finally, finalizing the diagnostic partnership was established late, causing the inability to meet the expectation for same-day co-approval, which can lead to uncertainty in decisions regarding the drug application. So as I said, the best way to prepare for such contingencies is to plan early. It begins with defining the biomarker and the technology that you will use to select patients, because basically the biomarker and the test will define your population. Start by selecting the biomarker analyte, whether it's protein, DNA, or RNA, and the specimen type, because CDRH will ask for analytical validation for each claimed specimen type, such as bone and blood marrow or formalin-fixed paraffin-embedded tissue versus frozen specimens. You should then select the technology. You will see that there are various pros and cons to each method, and you should consider how that will impact your trial population. For example, IHC may be very sensitive but have cross-reactivity issues. FISH may be sensitive but fail to detect all the fusions that you're interested in. Sequencing may have a wide variety of detection but fail to have an adequate sensitivity for certain biomarkers. So when selecting the technology, consider, is the ability or the capabilities of this test fully fulfill what you need in terms of defining the target population? I have seen, for example, where test poor reproducibility prevented the company from actually being able to bring in a companion diagnostic. And finally, select the cutoff or the clinical decision point, making sure that it's reproducible and that you will eventually be able to line that with the future tests that you bring in. So having selected your biomarker and technology, the next question that you should consider is whether you need an Investigational Device exemption, or IDE, for the use of the test in the trial. IDEs enable the use of investigational tests in patient management. One of the common points of confusion around testing used to enroll patients is that it's not investigational testing, because many times you are using these local labs and they're already running the testing and they don't consider this investigational testing. However, recall that I mentioned FDA regulates devices, including diagnostic tests, based on the specific clinical use. And in this case, it is to select patients who are expected to benefit or to predict response to the drug. In this situation, because the safety and efficacy of the therapeutic product are unknown, the safety and effectiveness of the test are also unknown, and so the test is investigational for this specific purpose. Whether you need an IDE is irrespective of the phase of the trial or the number of patients. So it's not uncommon to be confused and think phase 1s don't necessarily need an IDE or whatnot, but that's not the case. And it applies to U.S. sites testing foreign patients and foreign sites testing U.S. citizens. It does not apply to foreign sites testing foreign citizens. IDE requirements are also based on risks to patients. Sponsors have various responsibilities to mitigate the risks of these investigational tests so that they are safe for use. As clinical evaluation of investigational devices, you can take a look at this chart that I have here to determine the likely type of IDE study, device study that you have. All clinical evaluations of investigational devices, unless exempt, must have an approved IDE before the study is initiated. Exempt studies are those where results of the test are not reported to physicians for the purpose of management or treatment decisions, unless confirmed by a medically established procedure. All non-exempt studies fall into one of two categories: non-significant risk and significant risk. Both types have similar requirements. However, significant risk studies require the submission of an IDE application to FDA and approval of that application prior to the ability to use the test in the trial. A non-significant risk study is considered to automatically have an approved IDE, and provided that the sponsor follows the IDE requirements. So, how do you determine if you have a significant risk investigational study? There are several ways you can determine this prior to your trial. The first is that your IRB can make this determination. If you received a decision from your IRB, you do not need to make inquiry with the agency. On rare occasion, I should say, the agency may take a look at your study and realize that this is a significant risk and let you know, and if that occurs, the FDA's decision is final. Alternatively, you can submit a study risk determination request to CDRH. Study risk determinations are a type of pre-submission, and I'll provide you the link for that later. You would submit a copy of the clinical protocol for us to review. Alternatively, you can also make this request in the IND as one of the questions, provided it's an original IND. If you decide to ask the agency or CDRH, we recommend that you also include a brief description or explanation of why you believe the study is non-significant risk by answering 4 questions. Just a little bit about this. The agency recognized that patients who are enrolled into clinical trials are making the decisions with their physicians to accept the risk of the investigational therapeutic through the informed consent process, and these patients are heavily monitored for safety throughout the trial. Since the goal of an IDE is safety, patient safety, those devices used in this clinical trial are evaluated slightly differently than if the diagnostic was being evaluated on its own for another indication. The agency has determined it is appropriate to evaluate the risk of the device using these 4 criteria, which I'll describe. In essence, what is the risk to the patient, given that they've decided to go into this drug trial, if the test result was wrong? The first criterion is whether patients will be foregoing other effective treatments. For example, a patient with alternative effective treatment options may not have made the decision to participate in the trial if they did not have the biomarker. In this circumstance, a wrong result would cause the potential for the patient to forego a preferred option. Generally, many of these trials are in patients who have exhausted their options, and so a wrong result may not present a risk unless there is information that is known specifically about the biomarker subset, such as lack of effectiveness or safety concern. And that's the second criterion here. The third criterion is whether the trial will expose patients to adverse events worse than they would otherwise be exposed to in this clinical setting if they were continuing on chemotherapeutic regimens. It is very rare to encounter this criterion, but an example is when the drug is being used in combinations with drugs presenting a very high risk of toxicity. CDER or CBER may be determining that the study is safe to proceed because the test is identifying patients who have a very poor prognosis relative to the broader population. CDER may believe that the risks of the toxicity are acceptable for this population, but they would not be acceptable to someone without the risk, which is without this result, rather, which is why this is considered one of the criteria. Finally, is a specimen obtained solely for the purpose of testing the biomarker, and does obtaining this specimen expose the patient to the risk of mortality or severe morbidity in this clinical setting? I will say that a lot of people get nervous that they're going to need an IDE, but based on these criteria, easily less than 5% of the tests used in investigational therapeutic studies are determined to need an IDE application. And I want to point out really quickly again, because I think I said it already, these criteria do not apply to other types of investigational device studies. But if you find yourself needing an IDE, which again would be very rare, You should plan to use a single clinical trial assay, as noted in the earlier slide. But if that's not possible, then we recommend you use a central reference lab to confirm the results of all the other local testing sites. This enables you to send in an IDE for the one test that's being run at that lab. I would like to note that for non-significant risk studies, this is not recommended because it can create a layer of complexity for your future clinical validation, which I will get into eventually. Now, for some reason, as I said, sponsors are very reluctant to send in IDEs, and so I want to reiterate that IDEs are reviewed only for safety, which is basically that the test performs as expected around the cutoff. The focus is on data demonstrating that the clinical decision point is sufficiently robust for the proposed use. Data requested in an IDE is not at the level of an actual submission. It is generally maybe accuracy, precision, limit of detection, and we're very flexible with the types of specimens and depending on the stage you're at, so it's no cause for alarm. You should also make sure that the informed consent indicates that the test is investigational for the proposed use. So I have an example here. Let's see if you can quickly answer for yourself what you believe the risk would be. You are planning a first-in-human trial to explore the benefit of a drug whose tumor expresses elevated levels of a protein biomarker. You are using an IHC test to determine the expression level of the biomarker, and patients will be randomized to one of two arms based on their expression level. Archived specimens will be tested for the most part, and efficacy as a second-line indication is being explored. That means that there are alternative therapeutic options available to the patient. So what do you think? Is this a significant risk or a non-significant risk trial? Well, I imagine your first inclination is that this is a significant risk study based on these alternative effective options. But this would actually be a non-significant risk study because it's an all-comer study. Patients are not forgoing the alternatives based on this test result. They are being enrolled into the trial irrespective of the test result. I will add that even when it is not an all-comer study, medical officers are consulted, and if the alternatives, the clinical alternatives, are considered equipoise to the trial, that can frequently also render a non-significant risk decision. So you have your biomarker, your technology, and you are aware that you have a non-significant risk study. Next, you should plan to limit sources of bias that can occur based on your clinical trial testing strategy. While it is ideal to have a single test, it is not uncommon that sponsors are actually enrolling patients to a trial through the use of local laboratory testing or multiple clinical trial assays, particularly when much of this testing is already being done in the lab. So this can be problematic for the reasons that I'm kind of crudely illustrating here with this picture of the apples, which, let's say, represents your general population and your biomarker is green apples. The multiple different tests are going to identify the biomarker or green apples based on a lot of different features, different hue, different tints, different size, different amount of red. And so this discordance can lead to the enrollment population not reflecting the future companion diagnostic that you intend to bring. Also, pre-screening by local labs can create bias that will affect the success of your companion diagnostic validation by eliminating challenging cases, for example, and lead to inflated performance estimates for the companion. Basically, the risk with pre-screening samples is that cases that otherwise would have made it into the trial if your companion diagnostic had been used to select people did not make it into the trial with the laboratory tests, and you don't have outcome data on that. And this can be particularly problematic when you need to test these negatives in your bridging study and you don't have them and you're unable to estimate the discordance because you do not have the ability to understand the outcome of clinical trial assay negatives that would be positive by your assay. That is highlighted in this table here. When the test used in the drug trial is different from the version of the test intended for marketing, FDA asks that you conduct a bridging study. a bridging study is a statistical plan to assess the concordance between the results of the clinical trial assay and the companion diagnostic, and to assess the impact of this discordance on the original efficacy conclusions. The goal is to confirm that the companion diagnostic will support the selection of the same population and hence the efficacy of the therapeutic. So to accomplish this, you are asked to retest all the positive specimens as well as the negative specimens, or if the biomarker has a low prevalence, then a random sampling of negatives. Specimens should be annotated for factors that affect test performance and the factors that affect the therapeutic so that it can be demonstrated that missing samples are not biasing the conclusions. As discussed, though, if multiple clinical trial assays were used, you may not have test negatives. We always advise sponsors to ask the investigational sites to send forward some test negatives as well for screening, but frequently informed consent issues prevent that from being feasible. When that is not possible, we recommend that the pharmaceutical sponsor identify whether they can get specimens from another trial or similar trials. Finally, if that is not possible, we advise the sponsor to purchase specimens from the population. Frequently, the specimens are not annotated for the biomarker status, and so the negatives may need to be assessed by another method. And if that's the case, we'll ask that you send the procured specimens back to the labs that participated in the trial to identify the test negatives. It's not unusual for them to not agree to this, and so what you will then have to do is determine what were the most frequently used technologies used to accrue patients to your trial, and contract with labs who run those types of technologies to actually select negatives for you. For example, if there were 20 enrollment sites and the majority of patients were determined to be eligible by an NGS method, we would ask that a similar NGS method be used to identify test negative cases for your bridging study. You then have to estimate the discordance between your IVD and those negatives and impute that onto your efficacy analysis. So ultimately what I'm trying to illustrate here is that when you have multiple clinical trial assays and you don't have negatives with outcome data, which is to be expected, but if you're missing a large number of samples and you have a variety of discordance, it can get costly and it can complicate the analysis. And if the analysis using the market test results provides different conclusions from the original efficacy analysis, well, that will end up being a review issue, and I have seen that happen, and you don't want to be in that situation. So as mentioned, the ideal scenario is to be working with a single kit or single assay. That often is not possible, even if you intend it, because, for example, the biomarker under evaluation may be so rare that it would be prohibitively expensive to have a single site screening all subjects. So, in that case, you would want to attempt to have a central testing lab where labs can send patient specimens for testing, and then you will have access to some negatives. So, to explain, maybe you have you actually contract with one lab to be a facility where some labs will send cases. Not all labs will be doing the testing, and they will send you cases and they will screen, or you can encourage other labs to send forward negatives for consideration because your test is designed differently and you're trying to consider all options for individuals. If that's not possible, you should attempt to use sites that use the same technology. For example, if you have a fusion biomarker that you're using to enroll, you would want to select a single method for enrollment, such as IHC or FISH or the seq, not all different methods. Finally, you could attempt to qualify the labs to meet a specific threshold of performance so that you're not having different analytical sensitivities or the inability to detect certain fusion partners. You want to understand the prevalence of various fusion partners so that you can ensure that the tests are detecting the same one. Most importantly, you should collect information about all of the information in support of the local sites, include the lab name, the testing method, the analytes the lab has evaluated, and the prevalence of the biomarker in the lab. And be prepared to provide this information. There may be additional information that we request. You can work that out in discussions with us. Again, most importantly, attempt to get test negatives so that you can pre-plan the number of negatives for a bridging study. So by now, I've probably made it pretty clear that having specimens to support your companion diagnostic is critical. There are a variety of reasons why companies find themselves without specimens. So this is one critical plan, part of the planning, and that is to develop a sample acquisition plan. Plan to bank specimens from patients evaluated for and request adequate specimens to perform the analytical validation. For example, I have seen where the inclusion criteria may indicate a tumor block volume or a number of cut slide sections should be provided in order to enter the trial. Have a plan to obtain test negatives, for example, as mentioned previously, from a site established as a diagnostic partner. Consider obtaining paired specimens. For example, if you're enrolling on liquid biopsy and tumor, attempt to get both specimens. There are conditions that request. For example, the blood specimen should be obtained at the same time as the biopsy. Or if you're enrolling blood and bone marrow tests, you want to try and get some paired specimens because this is going to be necessary in the future validation. Consider the impact of storing specimens. You're going to be storing these specimens for the bridging study for a long time, and you should be evaluating the stability of the biomarker to ensure that you are able to test the specimens at the appropriate time with no or little loss in integrity of the analyte. We frequently see issues, for example, with cut slide stability and IHC assays. So you want to understand that your storing of these specimens will still make them available for testing. Try to have a single uniform method for specimen handling and pre-analytical steps. Otherwise, you're going to need to bridge that data. Plan for specimen stability studies in terms of the analytical validation by maybe collecting, aliquoting some of these samples and testing them at different time points. Consider policies in foreign countries. Some countries do not allow shipment of specimens outside of their country, so you will need to be able to test those specimens or account for them as missing in your bridging study. Ensure that informed consent allows for use of the specimen for test validation. That's another issue that we see. And finally, as I mentioned, ensure you have adequate annotation in the event you need to retest the samples and characterize them as being reflected, representative, of the overall test set. Percent tumor proportion, size of the tumor, percent necrosis, primary versus metastatic, biopsy versus fine needle. These are certain things that can affect test performance. And if you don't have all of these samples for retesting, we want to ask you to demonstrate that the specimens you did test are not biased and that they're representative of all the conditions of the specimens that went into accrual. As I noted, these should be supportive of the overall, these should be supportive of the representative of the valuable set for your bridging studies. So it is acceptable to explore optimal thresholds during the early studies, but it generally is not appropriate for the clinical validation study. This is another thing that we said early on that can be problematic. When planning the clinical validation study, the assay should be fully specified and locked down, and that includes all the clinical decision points and any scoring or interpretation rules for genetic biomarkers. So before you use any test on the bridging study, you should make sure that you have finalized it and you understand its performance. You should plan to ensure that instruments, software, and reagents that are for use with your test are legally marketed, as the agency cannot label assays for use with RUO instruments or reagents. This is also the case, I want to make a note, this is also the case for collection devices. If you have a liquid biopsy assay, you should make sure that you're collecting the blood with legally marketed tubes, or that you have a plan to bring that tube in with your assay. As noted, the companion diagnostic, the clinical validity for the companion diagnostic is supported by the drug trial. So avoid turning your validation set into a training set. This happens when you make changes to the test based on testing those specimens, because this will eventually mean that your study is no longer supportive of the clinical validation of the test. So the validation requirements are the same for companion diagnostics or complementary diagnostics. Complementary diagnostics are an alternative device type to develop successful therapies. Whereas companion diagnostics are those tests that are required for the safety and efficacy of the therapeutic, complementary diagnostic tests are those that stratify outcomes within the broader population. Complementary diagnostics are not a prerequisite for the safe and effective use of the corresponding therapeutics, but if response is reported in the drug label based on the results of a specific test, even when stratified, then this test would be considered a complementary diagnostic. Complementary diagnostics largely evolved out of the PD-L1 and PD-1 drug studies, where it was considered relevant to understand the differential response based on expression, but to not necessarily restrict the availability of the drug due to limited benefit. So the regulatory paths for complementary diagnostics are similar to that of a companion, frequently because you actually don't know where you're going to end up by the end of the trial. So you have to be prepared for both contingencies if you've enrolled an all-comer set and plan a retrospective analysis. There are many novel, unique drug review and approval paradigms by CDER and CBER that necessitate the need for novel companion diagnostic review strategies. So you will want to discuss your approach with the agency, but here's some examples. In cases where the approval is defined by the biomarker and not the histology, CDRH will generally ask for validation to include key cancer types that were in the clinical trial. However, the analytical validation will be based on those cancers where the biomarker is most prevalent, as well as those that are challenging, for example, thyroid specimens or melanoma specimens, because they represent, for example, small specimens with sources of assay interference. For cases where the biomarker might be denoted as a pathogenic variant in a structural rearrangement in a gene, for example, we ask for the detailed pre-specified rules for variant interpretation, such as gain or loss of function and specific details of how they are going to be interpreted to ensure that you have a definition that can be replicated after the trial. For liquid specimens, liquid biopsy specimens, where specimens are extremely limited and you have limited volume, we'll consider some contrived specimens. But we also ask that you pair them with resected specimens when possible for accuracy. We will frequently we'll use limitation statements and labeling that might refer to reflex testing also when appropriate to address the risk that, poor shedding or very low levels are false negatives, for example, in the patient population. Finally, all-comer patient enrollment strategies with biomarker stratification or with respective analysis with an assay has led to a number of scoring and reporting strategies with novel tests, such as TMB, PD-L1 immunohistochemistry tests, and the agency is working with stakeholders on how to harmonize these tests. So you would want to discuss with us very early on, if you plan on bringing in this test, your strategy. It's important to note that I've appended here on the side an image of a website that actually lists all of our cleared or approved companion diagnostic reviews and the summaries of all of the data that was submitted in support of those devices. So you can look at that. You simply click on the submission number and it will take you to the summary of safety and effectiveness, and you can get an idea of the types of validation that would be requested. So you have your test partner, you have a good specimen testing strategy in place. It's important to align the final review so that the test and drug can have the same day approval. The agency has experience with both laboratory developed tests and tests distributed by kit manufacturers. So either one of these we can work with. There are pros and cons to explore with each. I won't go into them, but for example, if you are unable to, if your instrument is research use only, a laboratory can assume responsibility for that instrument, effectively making it IVD, whereas if you have a distributed kit, in general, that's not possible. CDRH has a submission process akin to the CDER-CBER rolling submission process. It's called the modular PMA process, and it supports the timing of submissions by having the different device submission components submitted as modules. So GMP will be submitted as a module, software instrument can be submitted as a module, analytical can be submitted as a module in advance of the actual clinical validation data. Another option you might choose to consider is whether a humanitarian device or HDE application is appropriate. HDEs are basically used for rare disease. They're a little different from orphan products because they are based on the number of tests that are run per year, and that limit is 8,000. If you're interested in HDE, you can explore that with the agency. I generally don't feel that there's much advantage to an HDE because of the restrictions associated with them. and the clinical validation is automatically supported by the clinical trial. However, you might have another reason why you think that an HDE is appropriate for you. There's certainly benefits to the HDE program. In both cases, you will want to make sure that you allow time for manufacturing and BIMO inspections. You'll want to have letters of cross-reference from the diagnostic company and to the diagnostic company. And finally, if you want to keep your patient data private, you can submit it as a master device file outside of the diagnostic submission. The diagnostic company will refer to your data in the master device file, but they will not be informed of it or receive any deficiencies related to it. You would. All right, so how do you get feedback from us? We recommend that you obtain feedback from CDRH using the pre-submission process, which is a free interactive way to obtain information about study protocols, statistical analysis plans, or regulatory strategies, anything that you need help or feedback. We are motivated to work with you. We are not motivated to not let something get through, especially oncology drugs. Very excited to help you move those forward. They are, we will find a way to help you work. It's very important that you be open and honest with the needs that you have so that we can work together. A link to the guidance documents provided here. What you want to do is include your proposed intended use, a device description, and the specific questions that you have with the appropriate background that will enable us to adequately address the questions that you have. Briefly, I'd like to mention also the CDRH Breakthrough Device Program that may be of interest to some of you. This program is for certain medical devices that provide for more effective treatment or diagnosis of life-threatening or irreversibly debilitating diseases or conditions. If you're granted breakthrough device, it affords you an opportunity to interact with FDA more frequently on more recurring basis, and we prioritize the review of your submissions. You can look at the program for more information in the pre-submission guidance that discusses it. There also appear to be some potential reimbursable advantages to having breakthrough device designation, as some of you may have seen with the CMS notices. To date, I would have to say that many of the tests that are being deployed as original companion diagnostics in the oncology setting have qualified for this designation, so you might want to consider it. In summary, identify your biomarker and technology because this is how your target population is going to be defined. Define your clinical trial assay test strategy because, as I pointed out, this can complicate things depending on your timing and your ability to get specimens. Ensure adequate specimen collection for validation studies. I made the point that this is absolutely critical. And coordinate the submission timing. But I guess if I had to say what is the most important thing to your success, just one thing to do would be to engage CDRH early. It's very important to get the feedback that you need on and recognize the things that you're going to have to do in support of the companion diagnostic. In the next few slides, I won't go through them. I'll just tell you I've provided resources, different types of documents that can be helpful, especially and particularly I've highlighted those documents that refer to some of the information I've provided you here. The co-development guidance is very helpful. The study risk determination and the streamlined submission processes are highlighted here. And then eventually, and also there's more generic learning tools. And I also appended the reference to being able to ask for parallel review. You have to initiate this discussion on the end of the, with the payer task force, but it's maybe a mechanism for you that would be useful. And that's it. That concludes my talk, and I look forward to answering any questions that you might have during the question and answer session. Thank you very much.
Thanks, Donna. It's always a joy to listen to you talk. You're very direct. It's appreciated. This significant risk, high risk, significant risk determination thing has honestly persistently befuddled me for years. And I listen to you talk and I get it, right? And then 5 minutes from now, I'm going to get confused again. And looking at some of the questions, there's a bunch of significant risk questions there. So I'm going to hold my question off and run through some of these that are in the questions and answers. But one is, when would the requirement for a tumor biopsy collection be considered significant risk?
Thanks, Jeff. Yeah, so that's actually a really good question. Also, I see a lot of these questions are all really great questions. I'm impressed. One of the things that I want to note is that in the IDE regulations, having to use an invasive procedure to obtain a specimen is considered significant risk. But that wasn't practical in this oncology clinical therapeutic setting. And so we looked and said, well, given that these patients are undergoing surgeries and biopsies frequently as part of their standard of care, what are those procedures that are being done solely for the purpose of doing the testing that are unusual in that clinical setting, that expose the patient to significant risk? And I will tell you that during the evolution of this, we discovered that it was very challenging to pin that down because there was, factors that really weigh heavily into this, which might be the experience of the surgeon, the particular facility. So it's sort of a self-assessment that undergoes. And interestingly, some sites have opted to have an IDE because they feel that certain procedures are a significant risk. And others have said, no, this is not necessarily a significant risk. We generally look at those procedures that are, engaged, like pancreatic surgeries or, some of the mesothelial linings or whatnot. But a good example might be diffuse pontine glioma specimen testing in the pediatric setting. That is not part of standard of care to obtain those specimens. So in consultation with the oncologist at CDER or CBER, we will have discussions about the surgical procedures. So there's no blanket answer about the tumor biopsy. I will just say that it's very rare that triggers a significant risk decision because it's common for patients in that setting. So I saw that somebody else asked a question about the hematological setting. And if you were evaluating, a patient if you had an investigational test to say, I have a test that I want to look at to predict CML, you had to get a bone marrow specimen for that. That would probably need an IDE application for that indication. But in investigational clinical trials, it is very normal for CML patients to be continuously getting bone marrow, so we do not consider that significant risk, and that would not raise to the level of needing an IDE application. Hope I answered your
Thanks, Donna. You did. That's funny. The first thing that was coming to my mind was sticking a needle into the ponds, and you brought that up as well. My hope in the future would be that we have more and more targeted therapies based on science, and potentially those therapies are, as you know, many times, even though we encourage patients to have exhausted effective therapies, that sometimes in the informed consent document that we don't make them have exhausted effective therapies or even necessarily used every effective therapy or even a majority of them, but that it's kind of there. If a patient were to choose to go on to an experimental trial, we're not going to tell them that they can't. Sometimes, yet only 5% of these trials require IDEs. But if we're using that to enrich for patient populations and they haven't exhausted, and that word can be looked at different ways, then is it going to require an IDE? And then help us understand that most IDEs actually get approved anyways, right?
Yes, we generally, the bar for an IDE is not what you would expect it to be for an application. It's only reviewing the performance of the assay for safety, which means you have a clinical decision point for that assay. Do you understand how that's working? Is it robust for your purpose? Have you done the due diligence to mitigate the false results with that test? The idea is that patients who are going to be taking on unacceptable risks, having consented to the risks of the drug based on the test result, deserve to have an analytically validated test for that particular use. It's a part of the safety feature. In general, if the outcome of a wrong result is such that it would not have affected the person's decision to forgo that clinical trial and choose the well-known or publicized or publicly known effective option, then we don't require an IDE application because the risks are not considered significant. We understand through enormous amounts of patient engagement that CDER has had, that patients are very motivated to get onto trials, get onto targeted trials. They're willing to accept, risks, and there's no information known about that therapeutic as to whether the biomarker status makes an impact or not. In fact, that's been observed over and over again with different biomarkers, that in fact there's efficacy even in the negative population. It's just much lower, much reduced, greatly reduced. So those are the considerations we take into account in discussions with the CDER oncologist. What is considered clinical equipoise in this setting? And if the trial is considered clinical equipoise to those other alternatives, then the decision is that it's a non-significant risk trial.
Great, thanks. Another question from the audience. Please speak to whether it is reasonable to employ local labs for the dose escalation and initial signal syncing expansion cohorts, collecting tissues for respective assessment, then introduce a central assay as soon as expansion cohorts are initiated. That's kind of like, look, we want to see if the drug works before we start to go down this pathway. Is that okay?
So it's acceptable to use local labs. The point of the issue about the local labs is when you start to get to the registrational trial. That is when you start to introduce sources of bias and discordance and create potential issues with your future bridging study. But let's say, and I want to say that I have seen, very early phase study come forward because it was so encouraging, and now there's no specimens and there was local labs. And unfortunately, there's also a negative case I think goes back to like 2009 or whatever, I'm not sure, 2010 with Omipro, where they didn't have any specimens, and that was a bad outcome. So you do want to be prepared, although, obviously it's a case-by-case basis. What I was going to say is that the expansion cohorts are considered separately in the risk analysis. So, for example, I have seen scenarios where there's an all-comer study, and so no IDE is needed, but out of the initial portion of that, very toxic events were observed in the biomarker negative subset. And at that point, not only was the expansion cohort appropriate, but the data monitoring committee insisted that negatives not be enrolled. And at that point, a companion diagnostic became necessary. So we do consider the risks in the expansion cohort separately, but at the same time, this request. So just to quickly summarize, when you submit your study risk determination, either to CDRH through the pre-sub process or in the IND, and you've indicated you have an expansion cohort, we will consider that separately at the time based on the existing and available information. When you move into another phase, that is considered a separate trial, and you should submit another study risk determination for that trial, and we will consider the existing information at that time as well.
Got it. Thanks. Here's a question I think I can answer, but maybe I'm gonna let you answer it. We have a monoclonal antibody. The target is expressed in 96% of all solid tumor malignancies. They think it's going to have a low toxicity. Do we need to have a companion diagnostic for that?
So that is going to be dependent on if we agree with your assessment that it's present in the wide population. And I think I may be off on a different question. Was this question associated with the highly prevalent in solid tumors? Yes, right, yes. So if your biomarker is present in greater than 95% of the tumor tissues, what we generally recommend is don't screen and just enroll all comers, and then you don't need a companion diagnostic at all. If you feel that you must screen, then you're getting into that territory where safety and effectiveness might depend on the testing, and CDER will consider that. Look at those whether you have toxicity but if you don't, it's very unlikely that you would need a companion diagnostic, provided the label does not refer to any need for testing.
Thanks. I guess one of the ethical questions
I'll tell you a good example of that. Is the BRAF marker in Erdheim chest disease. There was no companion diagnostic needed for that because BRAF V600E is prevalent in over 95% of those cases.
No, I get that, and I understand. But an ethical question there would be, in the past we didn't have adriamycin. It works in a lot of tumors. It doesn't work in some. We don't have any marker as to why. Here, we can kind of almost have a marker. 4% of the patients aren't going to respond, but we're going to have efficacy in potentially other 96% that's going to drive those 4% that don't. Should we be excluding those 4% in the first place, right? And to me, that's an interesting question as to how we should approach that. I'm going to move on to another question, and I'm not sure I understand the letters here, but if a confirmatory CTA is used to confirm the presence of a patient selection biomarker that is intended to be submitted under a PMA, is a bridging study required?
Yes. So CTA stands for clinical trial assay. Yes. The question is the clinical trial assay going to be the test that you're bringing in as your companion diagnostic as well? If it is, then you're not going to need a bridging study. If it's not, then you're going to need a bridging study. And one of the things that I was pointing out in one of the slides is that if you have a central testing site confirming those local lab results, we're going to want the bridging study to go back to those local testing sites, not to the clinical trial assay. It's not an ideal. We have done that where it's compared back to the clinical trial assay, but it's not an ideal scenario and requires a lot of imputation analysis, statistical multiple imputations to deal with the missing samples and the bias and the prevalence, the bias created by the high prevalence due to the pre-screening in those central trial assays. I would just recommend ultimately that you, in the pre-submission process, you discuss your plan with us and your situation so that we can advise you appropriately on what you're going to need to do. But if it is the same as the companion diagnostic, no. And I want to go back to the issue with the 90% that you brought up, because you were saying, well, it's kind of confusing. It does seem confusing, but you have to realize there's certain error in every test, and the error in the test might be 5% or greater, then you're automatically introducing that error anyway into that population. So that's why if it's highly prevalent, we haven't generally required a companion diagnostic unless CDER believes that it is absolutely necessary for safety reasons.
Thanks, Donna. We got time for about one more question. This might have to make you take off the FDA hat, but you did allude to it. What happens if I'm developing a drug and it targets 0.1% of a particular oncology population, or it's an agnostic, and I'm trying to find patients across the world that have this particular translocation or point mutation or resistance mutation. Do you have any suggestions on how someone would do that?
Right. We've encountered that clearly with the NTRK, right? So, we recognize it would be extremely expensive to be trying to screen those patients yourself. What we ask is that you develop a plan to clearly collect information about the testing sites, the lab, the local testing that's being done for your accrual sites, your investigational trial sites. Collect information about the test, the technology, the specific performance, the prevalence of that biomarker at that site, the analytical sensitivity that they claim. You want to have a lot of detailed information about the tests. And then you will come forward with Excel spreadsheets, basically, for us, listing those tests, the number of patients that were enrolled at each site, and we will work with you to select 2 methods that you can bridge to.
Great. Thanks, Donna. We have run out of time. You have been a great colleague, and you're also a logophile with me, and I greatly appreciate that as well. And it's been a pleasure to have you give this talk.
Thanks, Jeff.